Hello, everyone. Distinct guests, professors, doctors, and everyone. Welcome to the symposium for today. So today, my topic will be on stem cells homing, unlocking the mechanism behind their therapeutic potential. So many people we know that stem cells is very important for a successful stem cell therapy is with their good therapeutic potential. So the therapeutic potential lies on what type of stem cells you have implanted. But what is most important is that do you know the mechanism behind this stem cells therapy? So this lecture I will be sharing with you all on the stem cells mechanism. How do they actually find their way home and give the regenerative properties? Okay, in general, the types of stem cells in the market are you can see they have embryonic stem cells and umbilical cord stem cells, adult stem cells, and of course, we have the fetal, progenitor, or precursor stem cells. However, different stem cells have their different abilities or different functions, such as the embryonic stem cells they are very pluripotent, means that they are able to regenerate into many types of cells, but they are potentially carcinogenic because if your body has some cancer cells, they might form teratomas. The other one is umbilical cord stem cells, which are obtained normally from the umbilical cord, or even the HSCs, which are obtained from the blood. But these type of stem cells are very limited, means that they are normally used for bone repair or such as for blood disorders. They are only limited capacity and limited therapies. However, another type of stem cells, which are the adult stem cells can be divided into iPSCs, or even from MSCs, or even some autologous blood-derived stem cells. This type of stem cells, they are based on the age, meaning that if you are 30 years old or 50 years old of the age from when you isolated the stem cells, meaning that you have older adult stem cells. Later in the lecture, I will explain why is it older stem cell not as good as younger stem cells. And the last one we have fetal precursor stem cells, which later on I will explain what is the beneficial effects of these fetal cells. In general, clinical applications for stem cell therapy can be used for regenerative medicine and can be used for blood-related disorders, which are limited to, like, for example, HSCs, and neurological disorders for the repair of or helping regenerative medicine in the terms of Alzheimer's disease, Parkinson's disease. And for respiratory disorders, the stem cells can be used for COPDs, musculoskeletal repair, such as bone repair, and cardiovascular repair. So these are the main clinical applications of stem cells therapy. In general, in this lecture, I would like to explain stem cells homing actually define successful stem cell therapy. Why is it so important? Because when stem cells are transplanted, they can detect the signals where are the places are injured inside your body. Which side they are injured or they have disease tissues or damaged tissues, the stem cells can detect the signals. And they will navigate through the bloodstream and migrate into specific microenvironments or the niches where they can continue to repair and regenerate your tissues. So I can say in this chapter, and stem cells homing is very important to ensure successful stem cells therapy. So today I will also explain what are the key steps of stem cells homing. There are 5 key steps in this topic. The first step is tethering and rolling. So once the stem cells are implanted to your body, the stem cells will start to, because when cells go into your body, as you know, your blood circulatory system is, the blood circulates very fast. But the stem cells will need to be slow, then they only can go to the targeted disease or damaged organs. So the first part is that the stem cells will go through like an amoeboid pattern, and they will start to, as you can see in these are the stem cells, and they will start to tether and roll, means that they will start to slow down. They won't be going very fast inside your blood circulatory system, and they will start to, they have the selectin and ligands. You can see the selectin from the endothelial cells of our blood vessels, and the ligands, the stem cells will bind to the selectin, so that the stem cells movement will be slowed. Second, once the stem cells have already been activated, the stem cells will be activated and they will communicate with the chemokines. The chemokines are secreted by the stem cells, and these chemokines will be coupled with the G protein, such as CXCL4, and this combination, this interaction will therefore activate the integrin receptors. Integrin receptors are important in cell adhesion. The third one is arrest. What happens next? The stem cells will then arrest on the endothelial membrane following activation of integrin VLA, which bind to the VCAM on endothelial cells. So as you can see, the stem cells has this integrin receptor which will bind on the VCAM, which is present on the endothelial cells. This interaction will cause the stem cells to attach very firmly to your endothelial cells. What's next then? Then is transmigration. The stem cells will therefore try to travel through the endothelial cell layer, and they will pass through the endothelial cells and go to the targeted organs. That's why this is called transmigration. And these are facilitated. How do the stem cells actually pass through? They will normally go through these MMPs. They will help to degrade and can pass through. There are two methods in general, stem cells, they will pass through and go to the targeted organs are by two methods known as transcellular and paracellular methods. The last step is migration. So stem cells will then migrate to the site of injury, guided by various signals released by the damaged tissue, such as growth factors and chemokines, and also cytokines. Once they migrate, and then they will go to your target organs, and the last step is that they will start with tissue regeneration and production. So these are the 5 key steps of stem cell homing. Just now I have mentioned the effective stem cell therapy, successful stem cell therapy is always based on a successful stem cells homing, which involves these 5 steps, known as tethering and rolling, activation by cytokines, cell arrest by the integrins, and transmigration, and lastly, migration, followed by the cellular regeneration and repair. And there are actually many journals which show that, because some people will ask, whether the stem cells will actually stay in the body after you have implanted the stem cells. There are actually a paper talking about this. They have actually transplanted the lung stem cells into the mice, okay? And then what happened next? So they did a tracking and checked that actually the lung stem cells did actually reside inside the lungs for up to 7 days. As you can see, the authors actually, the researchers have actually transplanted, they have actually tagged these lung stem cells with the fluorescence, and they have injured the mice with the naphthalene, and they found after 7 days, the stem cells actually, because this naphthalene will specifically injure the bronchioles of the rats. And as you can see, because I said the successful stem cells homing depends on the successful stem cells therapy. And because it is specifically injury on the bronchioles, then you can see that after 7 days, the stem cells home to the bronchioles instead of the alveoli. But the uninjured mice, which are the normal mice, the lung stem cells actually went to the alveoli instead of the bronchioles. What's next after homing? Paracrine signaling is very important because they are the nature's GPS for stem cells. Stem cells will release paracrine factors and followed by tissue regeneration and cell repair. And they are responsible for ensuring the precise location and homing of transplanted stem cells to their respective home cells. So once they reach the target site, the stem cells will induce the modification of damaged tissue microenvironment. And this paracrine signaling is very important, and because it will help cell survival, protection, immune modulation, and of course, tissue regeneration. There are two types of paracrine factors, known as soluble paracrine factors and insoluble paracrine factors. The soluble paracrine factors, which are produced by the stem cells, are such as cytokines, growth factors, chemokines, enzymes, bioactive lipids, and ATPs, UTPs. And the insoluble paracrine factors are the exosomes and the membrane-derived extracellular microvesicles. So I think from this lecture, you will know already stem cells can produce exosomes and also EXMVs. So what are exosomes? Exosomes are very small. They are 30 to 150 nanometers. They are cup-shaped, and they are usually released as excretory granules during exocytosis. And inside the exosomes, you contain a lot of proteins, which are very good for membrane transport. They can transport in membrane transport and fusion, and of course, heat shock proteins to prevent stress damage of the cells, integrins and tetrasplanins for adhesion. And in general, all cells mostly can secrete exosomes, which are very important for cell-to-cell communication. The next is membrane-derived extracellular microvesicles. They are bigger. They are around 100 nanometers to 1 microns. They are normally shed from the cells or surface of the stem cells by blabbing. And inside, they are like the membrane. They will package a lot of the contents, and they will blab out from the cells of the surface of the stem cells. And it contains a lot of proteins, cell surface receptors, metabolic enzymes, RNA transcripts, mRNA, and regulatory microRNA. And the release of the cargo contents will therefore continue to activate various cellular responses. So what do they do next? As in the previous slide, I have mentioned that the EXMVs contains mRNA. These mRNA are very important because they are involved in this transcription process, followed by translation process and protein synthesis. This protein synthesis then will be continue with cellular repair. Then transferred they will also contain microRNA, which they are important in gene expression. And they will be activated, downstream signaling pathways will be activated, such as PI3K pathway or MAPK pathway for cellular regeneration and tissue repair. So there are papers citing that the extracellular vesicles from the liver tissues, as I said, this paper, they have actually induced some toxicity to the livers of the rats by using the carbon tetrachloride, and the liver was damaged. Then after they implanted the extracellular vesicles and found that actually the extracellular microvesicles can decrease ALT, AST, and LDH. So we have already looked at the mechanism of the stem cells. How about what are the factors that will influence the homing of the stem cells? There are several factors which may influence the homing of stem cells, such as the stem cell source and type, what are the types of stem cell, and the age of stem cells, and what is the best route of administration of the stem cells. There are many types of stem cells which I have already mentioned in the second slide, and I will just briefly mention. In the market, there are mesenchymal stem cells which are normally obtained can be obtained from the umbilical cord or the fat or the bone marrow. And these stem cells can be differentiated into a few types of cells, such as osteoblasts, chondrocytes, adipocytes, and myocytes. And different types of these osteoblasts is for bone repair. Chondrocytes is for cartilage repair, and adipocytes for fat tissue, and myocytes for muscle repair. However, these mesenchymal stem cells have only limited functions, means that they cannot cure for every diseases. So they are only limited to these few functions. There are another type of stem cells which are known as the fetal, precursor, or progenitor stem cells, which we call as organ-specific stem cells, which these FPSCs can be isolated from placenta, thymus, neuron, brain, ovary, testes, kidney, artery, and eye, cartilage, heart, liver, lung, pancreas, pineal gland, pituitary gland, spleen, thyroid, adrenal cortex, mesenchyme, etc. And how do we actually isolate this type of stem cells? We do it by the primary tissue culture technique. So what do we use? How do we actually use this type of cells? Very easy, like cures likes. Hepatocytes used for liver regeneration. Cardiomyocytes for heart repair. So the comparison between like mesenchymal stem cells, they are only limited. But fetal precursor stem cells you can be using to treat a variety of diseases. The second factor, age of the stem cells. Do you think that age is very important? Or we can simply use adult stem cells to do implantation? So in this slide, I will share, aging actually affects the expression levels of function of adhesion molecules. When the cells are aging, the adhesion means that the stickiness, the cells are not as sticky as young stem cells. As you can see, Chambers et al. Said there is a decline in stem cells function and loss of epigenetic regulation in chromatin level of aging hematopoietic stem cells. And Liang et al. Also cited that there is an age-dependent decline of adhesion properties is observed between the stem cells and the stem cell niche. So they are not as sticky as young cells. Younger stem cells not only possess the ability to self-renew, but also have a greater differentiation potential. This paper is talking about how the impact of aging of regenerative properties of bone marrow, muscle, adipose-derived mesenchymal stem cells. And the study, they have isolated mesenchymal stem cells from 4 to 6 months old rabbits and also from old rabbits, 4 to 5 years old rabbits. And they did a comparison whether younger MSC is better or older MSC is better. from from bone marrow, from muscle, and adipose. And they found that the bone marrow mesenchymal stem cells isolated from the old rabbits have impaired proliferation and impaired chondrogenesis. And there is increased senescence in the aged mesenchymal stem cells. And all types of MSC, isolated from bone marrow, muscles, or even adipose, all the old MSCs have lower cell yield when they try to culture the MSCs. So from here, you will know that fetal cells are much better. Here, I'll show you a diagram. You can see if this is young stem cells, this is old stem cells. The cadherin complex and integrin complex are considered as the cell surface receptors of the stem cells. For young stem cells, they can bind more intact to our host cells, because you can see there are like more chains and these are lesser. So what do you think? Old stem cells are better or younger stem cells? Of course, the younger stem cells, because they have stronger adhesion properties. How about fetal stem cells? Why do we choose fetal stem cells as compared to adult stem cells? Fetal stem cells do not evoke any immune response, because there are many people asking, do you have any immune response after implantation? Fetal tissues do not evoke immune response because they express low or no MHC class 1 or 2. As you know, generally all our nucleated cells in our body expresses MHC class 1. And fetal stem cells also process higher binding interactions, increased adhesion properties, just like I say, the stickiness, and motility and facilitated transmigration across the endothelial cells, and go to the targeted cells and tissues. There are two examples here using the umbilical cord tissue-derived cells. They express low immunogenicity, and they have very low class 1 MHC, and they do not express any MHC class 2. And in general, mesenchymal stem cells, they are hypoimmunogenic, means that they will modulate the immune system and evade the immune surveillance. And they also express low MHC class 1. Okay, number 3, the factors, the route of administration. Which route of administration of stem cells are the best? There are many people, they thought that they will prefer to use direct transplantation or IV injection for stem cells. But direct transplantation, of course, it is very efficient because you directly target on that site and you directly implant. However, you might have, if you want to inject into the brain, which is very invasive, even though it is very successful in homing because you directly inject like brain cells to the brain, but very invasive and they may cause injury. So this is a very effective method, but a very, very invasive method. The second one is intravenous injection. They will just do IV infusion. However, this method, the stem cells will always be trapped in the lungs. Later, I will show a paper what is the difference between IV and IM. The third one is by intramuscular injection, which is a minimally invasive method, and they give promising results. They potentially treat both distant and systemic conditions, and they have longer dwelling time, means that the stem cells will stay in your body at a longer time, and it is safe. And there are alternatives such as intranasal application, means that you can directly intranasal, and then there are papers saying that it can help for certain brain disorders, but this is still under a lot of clinical it's still under a lot of clinical trials. So we still do not know how effective the stem cells will survive after intranasal application. So there are still many, many years to move forward to this approach. This paper actually tried to compare the survival and biodistribution of stem cells delivered via intravenous, subcutaneous, and intramuscular. Let's have a look. You can see IV infused, they are rapidly trapped in the lungs, and after a few days, in less than 7 days, the stem cells you cannot detect already. You can see these are the photographs, not called photographs, the fluorescence. After day 7, you cannot see any stem cells already. They have gone. Okay, subcutaneous, they are only detectable for 3 to 4 weeks. You can see, they still can see after 3 to 4 weeks. However, using intramuscular implantation, they have a longer survival rate until around 5 months. And they did until 104 days, and the stem cells are still present in the mice. And there's another interesting fact is that lower passage cells show optimal longer-term survival, meaning that, as you know, in the market, they are using mesenchymal stem cells maybe up to passage 5. So lower passage means that it's lower, like passage 1 or 2, is show longer-term survival of the stem cells. But our fetal precursor stem cells are primary, means that they are in passage 0, meaning that it can sustain a longer time and stay inside the body if compared to other administrations, mode of administration. So in conclusion, homing is the foundation of a successful stem cell therapy. And I have already mentioned 5 steps in homing is tethering and rolling, activation, arrest, transmigration, and migration. And paracrine signaling, which will have the paracrine soluble factors and insoluble factors, will enhance tissue regeneration and cell repair. The younger the stem cells, of course, the fetal stem cells, the stronger is the homing capability because they contain a stronger adhesion molecules, which they can successfully home to the targeted sites. And fetal precursor stem cells offer unique immune privilege because they have low class MHC class 1 or doesn't even have MHC class 2, making them very ideal for regenerative therapies. That's all from my side. Thank you.